Our next presentation is Verigraft AB, and I'd like to welcome Petter Borquist, CEO, to the stage.
Good afternoon, ladies and gentlemen. I'm happy to see still some brave souls here, willing to be entertained before the networking reception. So my name is Petter Björkqvist. I'm the CEO of Verigraft. I'm happy to, for the first time, be in Phoenix, Arizona. You know, my company is coming from Sweden, one of the Nordic countries, and it's kind of familiar to come here because, you know, in Sweden we are very often go to the sauna, and going outside here is right like going to the sauna in Sweden. Anyway, let's start. So, after many years of hard work, ups and downs, and a lot of preclinical testing, we ended up 2 years ago in this room in Granada, Spain. This was the day when we, for the first time ever, transplanted a personalized vascular graft to a patient. This was our first-in-man trial and a phase 1-2 trial I will talk to more about in a few minutes. This photo is not from a horror movie, it's from a patient in our trial. This is a leg of a patient with chronic venous insufficiency. This is a leg that is putting the patient into a really severe state of disease, into a really poor quality of life. So what are we doing and why are we doing this? This is another photo from another patient in our trial. On the left side, you can see the leg of this patient before we transplanted our personalized vascular graft. And on the right side, you can see the same leg 3 months later. I will, of course, come back to more details here. But the end of my initial set of slides here is taking you to the market. Our technology is a platform technology, so we can target a disease called CVI, that was the one I just showed a few legs from, and that is causing trouble for more than 4 million patients. I will come back to that. Arterial disease, another huge area of several indications where we are competing with some big companies on a mature market, today mainly occupied by synthetic grafts. Peripheral nerve grafts is another interesting area for our technology, rapidly growing. The last one, heart valve replacement. This is another huge area. You can see almost 20 billion euro we expect that market to grow to in 2031. So let's take a step back. We are in Sweden. We are a company being a university spin out from the Karolinska Institute in Stockholm. The photo you see here is from my office, and that is in another Swedish city, Gothenburg. We are a privately funded company. We have a highly experienced management team that has been around for more than 2 decades and made exit with similar companies in the area of regenerative medicine before. So I used to have one of these animals in my presentation. It's a lobster. Besides, I caught this lobster only a few weeks ago. It's really illustrating what we are doing, because a lobster, you can see here, this one is having one big claw and one small claw. And a lobster, if you break off a claw, can easily grow out a new organ, a new claw. But how about us humans? Can we grow out a new arm? No. You know what? That we cannot do this. Instead, we are hoping for other solutions for the future. And this is a bioprinter, as you can see. And I think we're all putting a lot of hope into bioprinters. I'm personally a big believer in bioprinting. But please remember, before we print hearts and kidneys and liver, that is taking probably decades before we do that at commercial scale. So what can we do? We can also do something good for the patients. We cannot produce new organs from nothing, but we can personalize organs. And I will tell you why we are doing that. So we are solving or targeting a huge challenge in medicine, and especially transplantation medicines. Besides that, there is a huge need for new organs and tissues. There is always also a real challenge with something else, and that is the immune system. We are transplanting hearts and livers and kidneys, but there is always a big but with that, and that is that we have to take care of the immune system, otherwise the new organ will be discarded, right? So we are doing that by using heavy immunosuppressive agents, and those come with a very high risk for several reasons. It's tough to live without an immune system. This is a lifelong treatment, but it's also a big problem because this group of drugs are connected to very severe side effects. To pick one of them, a dramatically increased risk of cancers. So what can we do? We work with personalization to overcome the problem with the immune system and the use of immunosuppressive agents. So now I will tell you how we do this. So every organ is built up by cells, but also something more. We call that a scaffold. This is a donor. This is a blue person here, illustrated as the donor. And in the other end of this figure, we have a patient. Let's say this patient is red. How can we go from blue to red? We do that in a series of first steps called decellularization. I think you know about that. That has been along for a long time. So we end up with a scaffold without any cells and DNA. And now this scaffold is anonymized. Then we do something that no one else does in the world, and that is to use blood from the patient. You can see that the blood from the patient we are targeting is going back in the system. And using the blood, we can personalize the scaffold in as little time as 10 days. And you should see that in comparison to many months that others are using when they are culturing cells, for example, stem cells. So we have a very cost-efficient, lean, and regulators that love this process because it is not involving direct manipulation of cells. So again, the whole idea here is to do this fully natural. We have something 100% biological. We don't need any immunosuppressive agents when we transplant because there is no reaction seen. So just one slide to show you that this is something really good, because on the left side you have a fully healthy blood vessel, a vascular piece of the vasculature, and on the right side you have our personalized graft. And as you can see, the cell content at the end are very similar, if not identical. So just to stop here and say that this part of the process, using the blood to personalize, is absolutely unique and covered by a really strong platform of IP. So back to the leg you saw in the first slides. The disease is called chronic venous insufficiency, and it's caused by the deep veins leading blood back from the legs to the heart again. As you can see, there is a system of valves that are regulating the blood and is sort of handling the enemy here, being the gravity, trying to press down the blood to the feet again. If those valves are failing, which they are doing for more than 4 million patients only in the US and Europe, you are in the state of chronic venous insufficiency. And this is a terrible disease, starting with a lot of pain and swelling of the leg, a feeling of heaviness, and ultimately this disease are damaging the skin and forming ulcers, bleeding, open, non-healing ulcers that are really putting the patients into a severe state. And this is a high unmet medical need. So unfortunately, there is nothing we can do for these patients today. So this is a little film from our product. So this is a personalized vein, and I think you can see the valve working in this piece of a vein. So again, our product is meant to replace a failing part of the vasculature. It's a fully biological, natural piece of the biology that will be fully integrated in the patient's body. So we are now in the clinic, as I said. We have a phase 1-2 trial ongoing in Europe, in Spain. We have, actually it's one more patient, so 80% of the patients are included in the trial. As you can see, it's a lot of attention in Europe from these type of trials. And to look again, this is a leg before surgery. After 3 weeks, you can really see how the skin is going to be repaired again. And after a few months, we have a completely new situation for the patient. So this is something we haven't seen before. We cannot treat these patients today with anything on the market. So this is just a statement from one of the lead PIs. So I mentioned this is platform technology, so we are working with other tissues as well. We are working with arteries, we are working with heart valves, and we are working with personalized nerves as well, the longer peripheral nerves. So there is one important thing I have to mention. As you saw from the first slides and the first product, we are using human donated cadaveric material as starting point for our manufacturing. This is very good, and when it comes to our first disease, it's needed because it's only us humans that are having valves of that type you saw here. But for the next generation of products, we are looking for some other raw materials as well. So instead of having humans as donor, we also see that animals can be donors. So xenogenic material, we are planning to use the food grade animals already handled today in industry. But interestingly, and you have heard quite a bit of that in this conference, we also look into the third step in this ladder. So we are working now on printing the starting material. So not bioprinting, printing because it's a cellular scaffold we are aiming for. So we are printing the scaffolds and then putting them into our proprietary process. So the day we can print, we can have an unlimited starting material supply. So coming closer to the end of my presentation, I'm here to tell you this story. I'm also here to tell you that we are raising funds now. We have something interesting ongoing right now. I have over the years been asked many times by private persons whether it's possible to invest and be part of this journey. But unfortunately, up to today, the answer has been no. But we have now started a crowdfunding to allow private persons to be on our journey. So we will, this crowdfunding will bring us a little bit into next year when we expect a clear new value step for the future. So when it comes to our product, the first product will be on the market in 2027, both in Europe and in the US. And we will, on top of that, or in parallel, leverage a portfolio of product candidates. So an exit is likely to happen in 2 to 3 years, about when we go to the market, I believe. So this is the crowdfunding. Should any one of you be interested, you can have a look at this one. I see at least a few cameras. Thank you. And you can also speak to me later in this conference. So let's end up with my lobster again. We are not trying to do new organs like lobsters, but we are trying to help patients in severe disease state. Thank you very much.